I hope everyone is fine. We are continuing the chapter 6, Prokaryotes. Today's objectives are role of bacteria in causation of disease and spoilage of food, bacterial diseases in man, bacterial diseases in plants, the bacterial flora of humans, benefits of the bacterial flora to humans, chemical and physical methods to control bacteria, and in the end, exercise. Now, the role of bacteria in causation of disease. Bacteria are useful as well as harmful to us. In our previous lecture, we have seen that bacteria are used in many different fields in the industries, in the synthesis and the formation or production of medicines in the food industry, etc. But there are many bacteria which are harmful to us, especially the parasitic bacteria, cause diseases in their host. Host may be a plant, animal, or even the human bodies. The parasitic bacteria that attack plants and cause many diseases, for example, fire blight in apple, ring diseases in potatoes, crown gall in number of different plants. And human diseases caused by bacteria. Diphtheria is infection in the throat and tonsils that leads to difficulty in breathing. Then heart failure, paralysis, and may lead to death. Tetanus infection is in the brain and nervous system, leads to stiffness in muscles, sometimes leads to lockjaw condition that results in death. Leprosy, it primarily affects the nerves of the arm, legs, and especially the skin, and cause sores on the skin. Meningitis is the swelling and inflammation in the meninges. Meninges are coverings that covers the brain and spinal cord. Sore throat, which is very much common. Whooping cough is a severe type of cough followed by a sound due to intake of breath. That sound is a woof-like sound. So that's why it is called whooping cough. It is also known as pertussis. Bacteria also cause decay of wood, leather, fabric, and also cause decomposition or spoil food, especially by decomposer or decomposing bacteria. Now, the bacterial diseases in man. Bacteria cause number of diseases in man, but few are given in your book. Cholera, Vibrio cholerae is the cause. Typhoid is caused by Salmonella typhi. Pulmonary tuberculosis by Mycobacterium tuberculosis. Pneumonia by Streptococcus pneumoniae. First one is cholera. Its symptoms are watery diarrhea, that is severe type of diarrhea. Cause is Vibrio cholerae. Treatment, of course, is replacement of water and electrolytes as the patient is dehydrated due to watery or severe diarrhea, and replacement of water and electrolytes is either orally or intravenously. Prevention is, of course, public health measures, and the supply of clean water should be ensured. This picture shows how the cholera spreads, that is due to contaminated water and food. And treatment is salt solution, which should be given intravenously or orally. Antibiotics are also used. As you can see in this figure, bacteria are dividing and multiplying in the intestine. Toxins are produced. That toxins cause cholera or diarrhea in the patient. Then typhoid. Symptoms are high fever, tender abdomen, and enlarged spleen. Cause is Salmonella typhi. Treatment is antibiotics. Prevention, again public health and especially the personal hygiene. Vaccine for the typhoid is also available. This picture shows the symptoms of typhoid: fever, headache, muscle ache, stomach pain, rash, especially on the trunk or on the abdominal part of the body, loss of appetite, constipation or diarrhea, and confusion, etc. Pulmonary tuberculosis. Symptoms are mild fever that lasts for 7 to 14 days, mild dry cough, night sweats, weight loss, anorexia. Anorexia means loss of appetite and weakness. Dry cough with blood sputum. Sputum is a discharge which is released after cough from the respiratory tract. If there is a blood in the sputum, this is a major symptom of pulmonary tuberculosis. Cause is Mycobacterium tuberculosis. Treatment: multiple drug treatment, which lasts for 6 to 9 days, 9 months, or DOTS. DOTS stands for Directly Observed Treatment Short Course, which is of 2 months. Prevention is a vaccine that contains Mycobacterium bovis, which is also called BCG generally. BCG stands for Bacillus calmette-guerin. This figure shows the symptoms of tuberculosis: loss of appetite, fatigue, chest pain, cough and prolonged cough, night sweats, pale skin, and this shows the infection which is in the lungs, or the site of infection is lung. Pneumonia. Symptoms are usually sudden chill, cough, and pleuritic pain. Pleuritic pain means pain in the chest. Sputum is red-brown or rusty in color. Cause is Streptococcus pneumoniae. Treatment: antibiotics are used. And this figure shows the main symptoms of the infection, especially pneumonia: high fever, chills, cough, shortness of breath, chest pain, fatigue, headache, loss of appetite, heartbeat increases, nausea, vomiting. And this figure shows these are alveoli. These are the alveoli of the healthy person, but due to infection caused by bacterium, that is Streptococcus pneumoniae, you see alveoli are filled with fluid, which then results in cough and shortness of breath. Now, the bacterial diseases in plants. The important bacterial diseases are leaf spots, blights, soft rots, wilts, and galls. First one is leaf spots. Symptoms: lesions on leaf blade, as you can see in this figures. Cause is Xanthomonas campestris on tomato, and many other genera of the bacteria cause disease in tobacco and potato. Prevention is to prevent the contact between the pathogen and the host, use of disease-free seeds. Blight. Symptoms are brown, yellow spots, withering, or dying of leaf, flowers, and fruits. Cause is Xanthomonas oryzae. It causes blight in rice. Other genera cause fire blight in pears and apples. Prevention, again, isolation of the diseased plant by physical method and the use of disease-free seeds. Soft rot. Symptoms: plant tissue die and rot-like symptoms on any organ. Cause is Arvina annulovora, that cause soft rot in potato. Cornybacterium cause ear rot in wheat. Wilting. Symptom is loss of turgidity in the leaf blades, which increase with time and leads to wilting and dying of leaf, as you can see in this picture. Cause is Pseudomonas solanacearum, causes wilt in potato. Prevention, disease-free seeds and spacing between the plants. Galls, these are localized outgrowths. It may be small or large. Cause is Rhizobium leguminosarum, causes small galls, which are also called root nodules in legumes. And the prevention is crop rotation and the removal of diseased plants by physical method.
Harmful bacteria. Some kinds of bacteria cause diseases in animals and plants. Mycobacterium tuberculosis, which causes tuberculosis. Vibrio cholera, which causes cholera. Bacillus tetani, which causes tetanus. Bacillus anthracis, which causes anthrax. Pneumococcus, which causes pneumonia. Haemophilus influenza, which causes whooping cough. Some diseases caused by harmful bacteria. Soft rot, fire blight, brown gall, etc. in plants are caused by bacteria. They also cause food spoilage, Staphylococcus, and food poisoning, Clostridium botulinum.
Now the normal bacterial flora of humans, there are approximately 10 times as many bacterial cells as human cells in the human body, and especially on the skin and digestive tract. They are useful to us, provide some nutrients, synthesize some vitamins for us, help in the stimulation of the immune system, and also prevents our body from the attack of other pathogenic bacteria. This figure shows good as well as bad bacteria. Good bacteria are those which are beneficial to us, and bad are those which cause diseases in man. This table shows the name of the flora and the location where they are normally present in humans. Clostridium species are present mostly in colon, Escherichia coli and other coliforms in colon, vagina and outer urethra. Lactobacillus species in the mouth, colon and vagina. Staphylococcus aureus in nose and skin. Enterococcus faecalis in colon. Viridans streptococci in mouth and nasopharynx. Now the benefits of bacterial flora to humans. The members of the normal flora, they are very important because they help in the maintenance of health and also some prevents us from the diseases. And especially, but although they are beneficial to us and is the normal flora of humans, but sometimes they cause diseases in man, especially the individuals which have impaired immune system, means weak immune system, and individuals are weak, feeble individuals. In them, that normal bacterial flora start causing diseases. Usually, this normal flora of the bacteria is non-pathogenic, but sometimes they are non-pathogenic in one part of the body, but when they enter in other part of the body, they start causing disease. And these non-pathogenic bacteria, especially they are present on the skin and the mucosa. Mucosa is the inner lining of most of the tubular organs, and especially the normal flora of the skin and mucosa It interferes with colonization, means interfere the attack of pathogens. The ability of members of the normal flora to limit the growth of pathogens is called colonization resistance. If the normal flora is suppressed, then pathogens may grow and cause disease. Intestinal bacteria, they produce vitamin B and K. The bacteria which normally live in the intestine, they synthesize vitamin B and K for us. This picture shows that this is epithelial cells and there is a mucus layer. This is the normal flora that is normally present in the human body. This normal flora prevents the attack of pathogens and limits the pathogenic attack.
Digestion is a complex process that our bodies carry out using a variety of chemicals. In this video, we're going to focus on the role of bacteria in digestion. Many different types of bacteria are found in the gut. In fact, scientists estimate that the gut contains between 300 and 1,000 different types of bacteria. And the total number of bacteria in one person's gut could be anything between 100 and 1,000 trillion. That's a lot of bacteria. So what are they doing inside our digestive system? The most common and important bacteria is the Lactobacillus species, which lives in our small intestine. When undigested, carbohydrates enter the intestines. Lactobacillus helps digest them, otherwise they would be lost from the body and unused. Lactobacillus also helps digest sugars found in dairy products. When Lactobacillus bacteria anaerobically respire, they produce lactic acid as a waste product. Although lactic acid in our muscles hurt, lactic acid in our gut is actually very useful because it keeps the pH of our gut acidic, which means pathogen bacteria, so bad bacteria, cannot thrive. This helps protect us from bacterial infections. Lactobacillus also helps prevent too much yeast growth in our guts, which if it spreads out to the rest of our body can cause yeast infections. Too many yeast cells can also cause problems with our digestive system. As well as aiding digestion, scientists have discovered that the bacteria in our gut can also influence our immune system, metabolism, and the production of essential biocompounds. There's even research showing that some cardiovascular disease, diabetes, inflammatory bowel disease, and obesity are associated with changes in our gut bacteria. But back to our gut bacteria and digestion. What happens if our doctor prescribes us antibiotics? Antibiotics work by killing bacteria or preventing them from reproducing. Does this harm our digestion and all the other wonderful things that gut bacteria do? Some antibiotics are targeted at specific types of bacteria, and so our doctors will try and target only the nasty pathogenic bacteria. However, often antibiotics will also kill our good digestion bacteria too. But if you're ill and you need the antibiotics, then this is just a side effect that we have to handle. There are a few things we can try and do to help our good bacteria re-establish healthy gut populations. Probiotics are live bacteria and yeasts that are found in some foods. It is thought that eating probiotics can help restore the natural balance of good bacteria in our gut that we lose from taking antibiotics. Only certain types of live bacteria make it through our acidic stomach, and they need to be in very high doses. Prebiotics are essentially food for bacteria. They stimulate certain bacteria populations to multiply and survive in the gut. Probiotics can be found in these foods amongst many others. So, while our bodies are brilliant at digesting food, it turns out they need a little help from friendly bacteria to get the job done, to catch any undigested food, and to maintain a suitable pH that we stay healthy, amongst many other great things.
If you like the video,
Now, the control of harmful bacteria by chemical methods. Bacterial growth can be controlled by chemical as well as physical methods. That is, chemical methods. Number of different methods are used. Number one, antiseptics. These are the chemicals used on living tissues to inhibit the growth of microorganisms. Disinfectants inhibit the growth on non-living material. For example, halogens and phenols, hydrogen peroxide, Potassium permanganate, alcohol, and formaldehyde, etc. are used. Number 3 is chemotherapeutic agents. They destroy the natural defense and stop bacterial growth in the living tissues. For example, sulfonamides, tetracycline, penicillin, etc. Now, the physical method to control bacteria. There are many different methods which are used to control bacteria. The most important one is sterilization. Many different methods for sterilization are used, including steam, dry heat, gas, filtration, radiations, etc. Sterilization is usually used to sterilize surgical apparatus and to preserve food items on the large scale, especially milk and meat, etc. High temperature is used in microbiological labs. Radiations below 300 nm are very effective, especially the gamma rays. They are used in sterilization. Heat-sensitive compounds like antibiotics, sera, etc., they can be sterilized by membrane filters, means they are filtered through membrane. Pasteurization is another common method of controlling bacterial growth. It kills microorganisms by heating at temperature enough to kill microbes. For example, milk is pasteurized by heating at 71 degree for 15 seconds or at 62 degree for 32 minutes. Food can also be frozen at -10 degree or 18 up to 18 degree for several weeks or up to several months. For example, meat, vegetables. Just low temperature prevents the growth of bacteria. Then dehydration is another method. Dehydration means removal of water from the food. It prevents microbial growth, especially dry milk and dry meat. Preservatives are the chemicals which are used and they inhibit microbial growth. Acid is also added sometimes that lowers the pH, and that pH is not suitable for the growth of bacteria. And in many cases, increased salt and chemicals like potassium metabisulfite, which are used as preservatives. They are used especially in the pickles, increased salt content, jam, high sugar content, and bread. They are preserved by the use of chemicals like potassium metabisulfite.
In this part of videos we will discuss about sterilization. Sterilization is the process by which an article is killing or removal of all microorganisms, including bacteria and their spores. There are two types of sterilization. First, physical method, and second, chemical method. Physical method of sterilization are done by dry heat, moist heat, radiation, filtration. Chemical method of sterilization are done by phenols, alcohols, halogens, aldehydes, and become red in the flame. Sterilization done. Hot air oven. It is an electric oven. Hot air oven is most commonly used method. It used for glass syringes, glassware, forceps. In hot air oven we take 160 degrees Celsius temperature for 1 to 2 hours. Moist heat. In moist heat we will start from pasteurization, most commonly the large quantity of milk denature by pasteurization. There is two method of pasteurization. First, holder method. In holder method milk stored at 63 degrees Celsius temperature for 30 minutes. And the second method is flash method. In flash method milk stored at 72 degrees Celsius temperature for 15 to 20 minutes. Boiling and steaming. For boiling and steaming we use 20% sodium bicarbonate water at 100 degrees Celsius temperature, for 30 minutes. Autoclave. In autoclave or steam under the pressure we use high pressure saturated steam at 120 degrees Celsius temperature for 15 to 20 minutes. Autoclave we done in a closed chamber, because the pressure of boiling water also raises. Radiation. There are two types of radiation: ionizing radiation and non-ionizing radiation. In ionizing radiation we use alpha rays, beta rays, gamma rays and X-rays. Alpha rays sterilization is best method for glass syringe, plastic syringe, and catheters. In non-ionizing rays are low energy rays. UV ray. UV rays radiation used for entryways, operation theaters, and laboratories. Filtration. Filtration is sterilization of heat-label liquid like serum solution, plasma solution, vaccine, antibiotic. This process does not kill microorganisms. It separates them. Second method of sterilization is chemical method. Chemical that can cause sterilization called sterilant. Some sterilant have very narrow spectrum of activity and some have very wide. Phenols. Phenols is first antibiotic to be used. Phenols are coal tar derivatives. Halogen. Halogen sterilants are oxidizing agents, like chlorinated, iodinated. Alcohols. In alcohols sterilants are 60 to 70% alcohols and water. Aldehyde. Aldehyde damage the protein. Surface active agents. Surface active agents they are divided into four main group. Number 1 cationic. Cationic has positive charge. Number 2 anionic. Anionic has negative charge. 3 non-ionic. Non-ionic has no charge. And number 4 amphoteric. Amphoteric has positive and negative both charge. This is all about sterilization.
That was all about the chapter Prokaryotes. Now come to the exercise. Question number 1. Select the correct answer. The option question and or the statement number 1 is that cyanobacteria are poisoned by oxygen, are not widely distributed, have chlorophyll, have chloroplast. The option is C. It's not given over here, but the option is C. Number 2, cyanobacteria, unlike other types of bacteria that photosynthesize, do A. not give off oxygen, give off oxygen, C. not have chlorophyll, D. not have a cell wall. Option is B. Number 3, pili are made up of pillin, which is A. carbohydrates, B. lipids, C. proteins, D. triglycerides. Option is C. proteins. Number 4, most pathogenic bacteria cause disease by A. directly destroying individual cells of the host, B. depriving the host of their nutrients, C. producing toxins, D. depriving the host of oxygen. Option is C. producing toxins. 5. Chemosynthetic bacteria are autotrophic, use the sun rays, oxidize inorganic compounds to acquire energy. Both A and C. Option is D, that both A and C are correct. Number 6, a bacterium with flagella all around is A. monotrichous, B. lophotrichous, C. amphitrichous, D. peritrichous. The option is peritrichous. 7, conjugation is facilitated by A. capsule, B. pili, C. flagella, D. both pili and flagella. Option is B. pili. 8. Bacterial membrane differ from eukaryotic membrane in A. Lacking proteins, B. Lacking lipids, C. Lacking polysaccharides, D. Lacking cholesterol. Option is D. Lacking cholesterol. Number 9. Bacterial membrane also contain enzymes for A. Respiration, B. Photosynthesis, C. Protein synthesis, D. Secretion. Option is A. Respiration. Number 10. Facultative anaerobes. A. Require a constant supply of oxygen. B. Are killed in an oxygenated environment. C. Do not always need oxygen. D. Are photosynthetic. The option is C. Do not always need oxygen. Facultative means they can live both in the presence of oxygen as well as in the absence of oxygen. Your assignment is read the chapter thoroughly and answer short questions. As I told you before, answers should be precise, to the point, and should not exceed 4 to 5 lines. That is all about the chapter Prokaryotes. Thank you very much.